Good morning, everyone. It's great to be back at London Calling. So I have no conflict of interest to report. Just to briefly give some context to why we're doing what we're doing. We're working on frontotemporal dementia. It's a neurodegenerative brain disease characterized by language or behavioral issues. And unlike Alzheimer's disease, it affects a different area of the brain. But other than that, it's characterized by neuronal degeneration and progressive dementia. And for that, we use Promethean genome sequencing to find variants with a potential role in frontotemporal dementia. We focus on structural variants, we focus on repeat expansions, and we've sequenced to now about 400 genomes from the human brain. And what I'm not talking about today is that we also find a novel repeat expansion in those brains. Happy to talk about that too. But methylation data comes for free with all what we're doing, with all the brain sequencing that we're doing. So I got distracted and stumbled on this side quest. So the other speakers have briefly talked about DNA imprinting before, but I'll introduce it more properly. So those are loci in the genome which have methylation that is dependent on the parent of origin of the chromosome. So depending on if you got a chromosome from your mother or your father, it is methylated or it is not methylated. So, and those have an effect on the gene expression in the locus. It's also known to cause disease if those are dysregulated due to mutations or deletions like Prader-Willi or Angelman syndrome. And there's a little cartoon showing that non-imprinted genes just show you expression of the transcript from both chromosomes, while imprinting for the maternal or the paternal chromosome will give you expression and methylation dependent on the parents that you inherited from. And these potentially have consequences for variants that are on the DNA. So if you just do short-read genome sequencing, you see variants. You don't know necessarily if they're on the expressed copy or not, and that might have different implications for association studies. And I'll walk you through some of the software that we have developed to look at methylation. And here is methplotlib with everyone's favorite imprinted gene, kinase. And it shows you here that in orange you have the kinase gene, which has expression from the H2 haplotype here. You see loss of methylation at the promoter site of kinase, and vice versa, the blue traces show you loss of methylation at the promoter of the kinase antisense gene. We also developed methylmap, which is aimed for more population scale visualization. So every vertical line here is an individual, or more specifically, it's a haplotype from the 1000 Genomes Project. And you see alternating purple and yellow places where there is methylation and absence of methylation from one haplotype to the next. So those are again kinase imprinted regions that show you at a large scale that this is quite reproducible across individuals. You can also use methylmap online. So we built it in a web app, and you can browse a lot of 1000 genomes data that have been sequenced with Nanopore sequencing in recent months. And you can, for example, in this case, look at the SNRPN imprinted region. Here is also hierarchical clustering activated. So you see nicely that there is in one color all those that are methylated, and in the other color all of those that are not methylated. So that appears to work quite well. It's quite reproducible. You see it in lots of samples. We really try to just not look at a single genome and establish what is imprinting and how does it look like, but how does it look like at the population scale in hundreds of genomes. And finally, our work in progress on imposter, our pipeline to detect imprinted regions, not just look at known imprinted regions and make some pretty pictures of those, but also just from the data itself, try to find out everything that is imprinted in the entire genome, and in this case, specifically in the brain. So the principle, I would say, is not too difficult. We look at regions of the genome that have a large absolute difference in the methylation. So you compare haplotype 1 with haplotype 2, and then you see if there's a large difference and see if that's the case across many individuals. And then we combine those positions vertically into regions to see, well, what exactly is here different between haplotype 1 and haplotype 2 across all the individuals that we're looking at. So essentially, we start with Dorado base calling and modification calling. We do minimap2 alignment to the reference genome like everyone else. We use longshot for phasing the data, although you could equally well do it with WhatsApp or longphase. We do mod-kit pileup per phase to get the methylation frequency per haplotype 1 and haplotype 2 for everyone. And then our imposter snake make workflow starts. It starts with just reformatting, splitting, joining, concatenating, and sorting. It takes a while to get all the data in our preferred format, but then we can start calculating the absolute difference between both haplotypes and then gathering those across many individuals. We throw out some outliers in the process with an isolation forest method, to just make sure that we don't have any too special cases that change too many positions in too many individuals. And then we filter the positions for those that have a shared large absolute difference between haplotypes, merging neighboring positions into intervals, and finally end up with a list of imprinted regions for the visualization. So this is a subset of the data that we get. So you see on the x-axis the percentage of values that have been missed in this analysis. So there's always some repetitive regions that are only seen in a few individuals that don't give Q methylation in everyone. And on the y-axis, the percentage of values that are highly different between haplotype 1 and haplotype 2. And most of the data is at this very spot in the bottom. So most of the individuals have a call here, and for most of the individuals, it's the same between haplotype 1 and haplotype 2. Most of your genome is not imprinted at all, but those are the ones that do interest us. So those are the ones that are we have information for many genomes, but for most of the genomes, it's highly different between one haplotype and the other one. And with this method, we could already identify 42 imprinted loci. So it's slightly less than expected, but all of them are well-known imprinted loci. So at least it shows that the method works. And for some visualization, here is GNAS again. Now in red, you see here the absolute difference between haplotype 1 and haplotype 2 with hierarchical clustering on the rows. So most of the individuals indeed show the expected imprinting in these positions. Other examples, like recently discovered loci like NAT1, shows similar results. And also, oh, that was my last example, shame. But some more things to be done for imposter include further fine-tuning of our pipeline. We're sure that it's highly specific, but could probably be more sensitive. Happy to get some feedback on that. We would like to add trio data to just not know that this is an imprinted region, but also to be able to identify, well, this is maternally or paternally imprinted. We will integrate it with lots of transcriptomics data that we have from the same brain individuals to also identify expression differences to see what is now, which imprinted loci differently has an effect on gene expression or transcript expression, and overlay this with GWAS hits. So if a locus is associated for a GWAS, then having the methylation information could be very important. So with that, I would still like to thank those people that have been involved in this project, with those in red boxes present here in the audience. And I would also like to acknowledge all our collaborators who send us samples from all over the world, from rare disease cohorts, to make our work possible. And with that, I would like to thank you for your attention.